Hi ladies, in case you'd like an audio-visual style of revision and would like to see a few things talked through, this is Mr. Heath here with a little bit of revision for genetics and evolution. I'll go through some key points, some very useful points, not all. I may give a few hints here and there, but nothing earth-shattering, but interesting to listen to all the same. So we'll start with genetics. So our first stop on the genetics road is DNA. Now, it's important that you know that the four bases in DNA are adenine, cytosine, guanine, thymine, ACGT. The letter is sufficient, and you must know that adenine always pairs with thymine, cytosine always pairs with guanine. What's also important to remember is if you look at the backbone of DNA, it consists of alternating phosphate sugars, phosphate sugar, phosphate sugar. Okay, and it's important to know that this backbone here is made up of phosphate sugar. That's why we call it the phosphate sugar backbone or the sugar phosphate backbone. And it's also important to know that the sugar is what actually attaches to the base in the middle here. Okay, so these four we call nucleotide bases. Okay, so you have a phosphate sugar backbone and it's the sugar that attaches to the nucleotide base. And that whole unit there of the phosphate, sugar and base we refer to as a nucleotide. And of course DNA wraps itself up into long strands that are all clumped together called chromosomes, and we tend to see, and we always see DNA in the nucleus of a cell. We'll talk more about chromosomes when I get to mitosis and meiosis. So another thing that's really important to know is how to sequence. Okay, so you know that adenine, cytosine, guanine and thymine are the four nucleotide bases that we find in DNA, but it's also important to remember that DNA is a two-stranded substance. Okay, so we have, for example, a template strand and the complementary strand of that. And you need to know that A and T match up and that C and G match up. My little hint to help remember that is just to write them out in alphabetical order and think outsides match together and insides match together. So whenever you have a template strand, such as this one here, ATA, GCA, TCA, the A here is always going to be complemented by a T. The thymine here is always going to be complemented by an adenine. Just like over here, the cytosine here is always going to be complemented by the guanine here. Okay? It's important to know that because you may need to actually sequence DNA. You also may need to understand why we can have different mutations and so forth that will change that sequence. Okay? Remember that a gene is a sequence of bases along a chromosome. So you might have 1,000 sets of base pairs, which makes up one particular gene. Okay? And that gene codes for a particular type of protein, which then codes for a particular trait or physical characteristic. Doesn't always work though. Okay? We can have what we call mutations that occur in the sequence. So a mutation is an error in the sequence of bases that make up a gene. So for example, if our original was GTA, CGC, there might be an error that happens when we're trying to replicate DNA in mitosis or meiosis, and instead of GTA, the copy gets GTG. This we refer to as a substitution mutation. It's also possible to have an insertion mutation where you get an extra base inserted into the sequence by mistake, or a deletion mutation where you actually get one that's just chopped out and everything slides up. A mutation only affects your phenotype, your physical characteristic, if there is a change in the protein that's produced as a result of that mutation. Don't forget also, sometimes mutations can have no effect whatsoever. Sometimes they can have a positive effect, and sometimes they can have a negative effect, which becomes really important when we talk about evolution later on. The other possibility here is something called a chromosomal abnormality. This is where there's an error in the number of chromosomes that an individual has. So for example, normally when chromosomes get passed from parent to child, we receive 23 chromosomes from mum, 23 chromosomes from dad. What can happen is that by mistake, we might receive 23 chromosomes from mum, but receive 24 from dad. So a really famous example of that is Down syndrome, otherwise known as trisomy 21, which I'll show in a moment. But chromosomal abnormality is an error in how many chromosomes we have, so we might have 45 or 47 rather than the normal 46. A mutation is when the bases within DNA are actually changed in some way in terms of their sequence. So in order to look at the arrangement of chromosomes that an individual has, we use karyotypes. What we do is we capture a cell in metaphase and arrange all the chromosomes into an order where we can see all the different ones that we have. So you can see this is an example of a human karyotype which has all the 23 pairs of chromosomes that exist. So don't forget, we have autosomal chromosomes, which we number 1 through to 22, and those are identical whether we get them from mum or from dad. And then the 23rd pair of chromosomes are these sex-determining chromosomes down here. So the options obviously there are either X or Y. If you're XX, you're female, XY is male. We can use karyotypes to look at chromosomal abnormalities and to look at some genetic disorders. So if we look at Down syndrome as an example, you can see in the karyotype highlighted on the screen that on chromosome number 21, this individual actually has 3 copies of chromosome 21. And this is something that we call trisomy 21. Trisomy basically means that there's 3 of that chromosome rather than the usual 2. There are other examples which you would have looked at in class, things like Turner syndrome, where you only have one X chromosome and no Y chromosome. Okay, so you're missing a chromosome in that particular case. We would expect you to be able to look at a karyotype and determine whether or not it's a normal healthy karyotype, in the sense that there's the appropriate number and combination of chromosomes, or whether there's an abnormality like this one here. Equally, you might be asked to determine the sex of an individual from the karyotype, and if you were to look at this karyotype here, you'd be able to say, well, that's clearly a male because there is an X chromosome and a Y chromosome. XY is male, and so forth. Let's move on and talk about some terminology here that's very important. It's absolutely crucial that you understand the difference between homozygous and heterozygous. When we say that an individual is homozygous, it means that they have 2 copies of the same allele for a particular trait. So for example, if we're talking about brown eyes and blue eyes, if someone has 2 copies of the brown eye allele, so they received the brown eye allele from mum and the brown eye allele from dad, then they are homozygous for that allele. If, however, they have 1 copy of the brown eye allele, perhaps from mum, and 1 copy of the blue eye allele, perhaps from dad, then they are heterozygous. Remember too that when we're talking about an allele, we're talking about a different version of a gene. Okay, so we have one version which codes for brown eyes and one version which codes for blue eyes. Another set of terms that's very important to know the difference between is dominant and recessive. So when we say that an allele is dominant, what it means is that this is a characteristic that will show if the individual has at least 1 copy of that allele. So in the brown eye, blue eye example that I just mentioned before, brown eyes is dominant over blue eyes. So that means that if you have at least 1 copy of the brown eye allele, that you will show the brown eye characteristic. Okay, so it doesn't matter if you have just 1 copy of the brown eye allele from mum, or you have the brown eye allele from mum and the brown eye allele from dad, you'll show as having brown eyes. That's what it means to be dominant. But there is a difference in how we would describe it in terms of the homozygous heterozygous. So if, for example, someone had the genotype R, they're going to show the dominant phenotype because they have at least 1 R. But this individual, we would say, is homozygous dominant because they have 2 copies of the dominant allele. In contrast, we would say that this individual is heterozygous because they have 1 copy of the dominant allele and 1 copy of the recessive allele. A recessive allele is one that codes for a characteristic that will only show if that individual has both copies of that allele. So in this case, little r. So if someone has a recessive genotype, they can only be homozygous recessive. That means 2 copies of the recessive allele. So if you were to see a question that were to describe for you a cross between a heterozygous male and a homozygous recessive female, that would tell you that the heterozygous male is going to be 1 copy of the dominant allele, 1 copy of the recessive allele, and the homozygous recessive individual will have 2 copies of the recessive allele. So let's talk about how we actually get these alleles from our parents. There's 2 types of cell division that we've heard about. One is mitosis, one is meiosis. This is just a general table to give you an idea of the differences. We expect you to understand what the differences between mitosis and meiosis are, where mitosis and meiosis take place, and what the result is. So if we're talking about mitosis, what we're talking about here is cell division that occurs just for general growth and general repair of cells. We start off with one parent cell, and at the end we finish up with two diploid daughter cells. Diploid means that there is a full set of chromosomes. So at the first cell, we started off in a human cell with 46 chromosomes, or 23 pairs. And at the end, we finish with two diploid cells that each have the full 46 chromosomes. So in this case, we would say that the diploid number is 46. In meiosis, things are a little bit different. So first of all, meiosis really only occurs in sex cells. So for example, for the production of egg cells and sperm cells. We start off with the one parent cell, same as mitosis, but the end result is that we have two sets of divisions, so we get 4 daughter cells at the end, but DNA has only replicated once. So each of those 4 cells at the end has a half set of chromosomes. So whereas the parent cell had 23 pairs and 46 chromosomes in total, each of the daughter cells after meiosis has 23 chromosomes, exactly half the full number. So if you see an example of cell division that ends up with 4 cells, you know that that's going to be meiosis. You know that all those cells are going to be haploid and have only half the full set of chromosomes. If you see a type of cell division that ends with 2 cells, you know straight off that it's mitosis. Now, there is a reason that I'm discussing mitosis and meiosis here. The reason is that when we talked before about homozygous and heterozygous, dominant and recessive, we know that each individual has 2 copies of a particular gene. So if we're talking about the brown and blue eye alleles, each of your parents has one allele that they receive from their mum and one allele that they receive from their dad. Then they have each passed on to you one of those 2 alleles that they have. And the reason that they only pass on one is because in meiosis, DNA is separated out such that only one allele for each characteristic is passed into the egg cell or the sperm cell. Then when the egg and sperm cell fuse, you have one copy from mum, one copy from dad. So if we look here at a Punnett square, which can break down the possibilities of the alleles that an offspring can receive, what I've got here as an example is a cross between 2 heterozygous individuals. So we have a heterozygous parent here who has one copy of the big G allele, one copy of the little g allele, and a second parent here who is also heterozygous and has one copy of the big G, one copy of the little g. So what can happen is that there's a 50% chance that this individual passes on the big G to their children. 50% chance that they pass on this little g to their children. And likewise for this parent here. So what we end up with is 4 possible combinations. In this case, we can have one offspring who has big G, 2 possible offspring here and here who have one big G, one little g, so they're heterozygous, and one possible offspring who has the little g, so is homozygous recessive. We'll often ask you to write out a genotype ratio here, in which case you must include the genotype. So you must say 1 big G, 2 big G, 1 little g. You can't just write 1 to 2 to 1 because you'll be marked incorrect if you do. But we also can't forget here as well that whether you have big G or big G, as long as you have one copy of that dominant allele, you will show the dominant phenotype. So here in the phenotypic ratio, we have 3 green arms, if big G codes for green arms, and only one individual, the one who's homozygous recessive, who will have white arms. That's the end of part 1. In part 2, I'll move on to talking a little bit more about alleles and X-linked inheritance, as well as moving on to evolution as well. Stay tuned for the next one.